Hello, in this video, we are going to be talking about the concept of antimicrobial resistance. So resistance is a growing problem around the world where these bacteria have become resistant to certain drugs. So when we give them the drug, they don't work anymore. And obviously that is very scary and a growing problem. So in this video, we're going to talk about how that happens and what these bacteria do to become resistant. So how resistance occurs, it is essentially a natural process. So when we think about living things, when you try to wipe out an entire population, there's always going to be some quote unquote survival of the fittest. So just like how, most organisms through evolution are going to try to survive, whenever we try to give antibiotics to a patient to kill off these microbes, every once in a while, you're going to get a random mutation or something where the microbe is then resistant. And of course, that's going to be a problem. So this is just a simple scenario of how antimicrobial resistance can occur. So if you were a person who is sick with some kind of pathogenic bacteria, and you take antibiotics, what you're doing is killing off what we call the normal microbiota. So the normal microbes that live in your body. And usually these microbes, they don't harm you, they're just hanging out. And they actually help with keeping any kind of pathogenic bacteria or resistant bacteria in check because your normal microbiota typically grows more than the pathogenic. Now, when you take a broad spectrum antibiotic, which basically kills everyone, everyone dies. But let's say that the pathogenic bacteria just so happens to be resistant to that drug that you're taking. Not even completely resistant, even if they were just moderately resistant. And let's say you only took one dose of your antibiotic, that bacteria then can survive and grow more because everybody else, all the competitors, are now dead. Now, if you were prescribed antibiotics for multiple days, usually they give 5 to 7 days, most of the time you will then also kill off these microbes, especially if they're not 100% resistant. Let's say they're just moderately resistant, then they would eventually die as well. So this is another reason why it's important to take the entire antibiotic dose. So if a doctor prescribes a patient 5 days or 7 days, they should finish it because that ensures that we are killing off any moderately resistant microbes. But this is essentially how resistance occurs. And again, it is a natural thing. Every time we take antibiotics, every time we use antibiotics, there is the risk of developing a version of that bacteria that is resistant. And again, this is just nature, survival of the fittest. Now, how exactly do these bacteria resist antibiotics? Well, there's different mechanisms. So one way is that they decrease their permeability. And so the drug just doesn't enter the cells. This could be a modification to the cell membrane or the cell wall, but essentially the drugs just don't get in. Now, if the drugs do get in, some bacteria produce efflux pumps that essentially just push the drugs back out. And so same thing, if the drugs can't get in or if they're pushed out, then they don't work. Some bacteria can modify the target of that drug. So for example, if we have a drug that targets the ribosome or the cell wall, whatever, if that antibiotic targets something and that target happens to be modified in a way so that the drug doesn't actually bind to it or affect it, then of course the bacteria is not resistant. And then finally, there are some bacteria that can produce enzymes that actually degrade or inactivate the antibiotic. So for example, some bacteria produce enzymes that break down beta-lactams. So all beta-lactams don't really work on these bacteria, which obviously is unfortunate. And what's really scary is that some bacteria are what we call multi-drug resistant, meaning that they have multiple mechanisms of resistance. So they're resistant to beta-lactams, but they're also resistant to the cephalosporins, and etc. So, they're just resistant to a lot of things, and that would be obviously problematic. Okay, so there are different ways to measure a bacteria's ability to resist a drug, and this is a lab technique that is done to, basically figure out how resistant is a bacteria to a specific drug. And that's going to be really important in terms of, how much are we going to prescribe, what kind of antibiotics should we prescribe, etc. And so before I start with how this works, let's talk about this word called susceptibility. It is essentially the opposite of resistance. Susceptibility means how sensitive the bacteria is to the drug. So if a bacteria is very susceptible, that means that they are killed very easily to that drug. And so in this diagram, I'm showing what is called the minimum inhibitory testing test. And so what we're doing is growing bacteria in different levels of antibiotics, usually from a low level all the way to a really high level. And we look at the bacteria's ability to grow in the presence of that antibiotic. And the lowest concentration that prevents the growth of the bacteria is called the minimum inhibitory concentration. So for example, if we look at these test tubes, the bacteria is able to grow at 2 and 4, but not at 8, 16, or 32. So we would say that 8 is the minimum inhibitory concentration, the MIC, because that is the minimum concentration that inhibits the bacteria. And same here, you can do it in a tube, or you can do it on a microplate, which has multiple smaller containers. And here you can see that they're testing different types of antibiotics for one type of bacteria. And again, same thing. It's the lowest concentration that prevents the growth is what we're going to call the MIC. And again, that tells us a lot about how resistant the bacteria is to that drug. And then there is the disc diffusion test. So sort of similar, we are testing how resistant the bacteria is to a drug. So what we do is we get a petri dish and we spread the bacteria on the surface of this petri dish so that they form a lawn. So a lawn is basically where the bacteria is growing all over the whole plate. But before we incubate it, we're going to add these antibiotic discs. And when the disc gets on the agar, it starts to release that drug. And there's different discs for the different drugs. So for example, you might have a disc for penicillin, and then another disc for tetracycline, etc. And so you lay those discs out, and then you incubate the whole thing. And what's going to happen is the bacteria are going to grow as a lawn, where they're going to cover the whole plate. But if the bacteria are killed by the antibiotics, then of course they're not going to grow. And they're going to create these little circles where there's no growth. We call those circles the zones of inhibition. So all of these regions where there's no growth is called the zone of inhibition. And you basically just get a ruler and you measure the diameter, and that kind of tells you relatively how resistant / susceptible the bacteria are to that antibiotic. And the general rule is the smaller the zone, so the smaller this diameter, the more resistant. Because if you think about it, if the bacteria are very resistant to that drug, they'll be able to grow close to the disc. Remember that the closer you are to the disc, the more drugs there are, more antibiotics there are. And so the bacteria are able to basically grow really, really close to the disc, and they're more resistant. Now, if your zone of inhibition is very large, that means that the bacteria is not as resistant. In other words, they are susceptible. All right, so that was antibiotic resistance. I hope this was helpful, and I'll see you in the next video.